Alright, so today we're going to talk about mitosis and the various stages of mitosis. Before we get started in the different stages, it's going to be important to understand the role of mitosis in reproduction. So reproduction is going to be making more of something. Mitosis is a type of asexual reproduction. So what that means is that it does not require genetic information from two separate parents. But rather it's just one single parent producing two. This is different from sexual reproduction, which is what we'll talk about in the meiosis video. The purpose of mitosis is to create diploid cells. So what does that word diploid mean? Well, that means to create an organism that contains a full set of chromosomes. And the types of cells in your body that are diploid are known as body cells or somatic cells. And some examples of body cells or somatic cells are things like skin cells or liver cells or heart cells, anything that is not a sex cell. So we're going to go through and kind of talk a little bit more about those different stages. Let's first talk about chromosomes and DNA in eukaryotes. So the DNA in cells is actually pretty interesting. If you were to take DNA out of one of your cells and stretch it from one end to the other, it would be more than 2 meters long. If you were to take all of the DNA out of all of your cells and stretch them out from one end to the other, you would have enough DNA to go back to the sun 250 times. And so that DNA has to be highly organized within your cells. And we typically talk about that organization as chromosomes. But let's start a little bit lower than that. So you have the basic structure of DNA, which is a double helix structure. That DNA is going to wrap around these proteins called histones. A way to think about this is if you had beads on a string. This is how it normally exists in cells in the form of what's called chromatin. This is just DNA wrapped around one of those little balls of histones. When we have several histones clumped together with DNA wrapped around them, you get what's called a nucleosome. And those nucleosomes can start to coil around each other to form this structure here. And then those coiled nucleosomes will then supercoil to make it even more condensed. And that is what you actually see when you see a chromosome. So let's break down what the overall structure of a chromosome is. So a chromosome consists of two identical chromatids. These are called sister chromatids. Remember from DNA replication that one DNA, so if this is a strand of DNA, is going to make a copy of itself. You now have a chromosome. It has two arms. The P arm is going to be the shorter end of the chromosome. It's not always the top part, but in this diagram it is. And the Q arm is going to be the longest arm in the chromosome, and here it's on the bottom. And then this is just showing you that DNA molecule kind of unwinding based on this diagram. So let's start talking about the different phases. This is going to be fairly quick. Prophase, this is the first phase of mitosis. This is when that chromatin that was just wrapped around histones or beads on a string starts to supercoil and form chromosomes. We have these structures called centrosomes, which contain a pair of centrioles, so that's these structures here. They start to move to opposite ends of the cell. And then the nuclear membrane begins to break down. Here's a schematic here, and this is what a microscopic image would look like. We then have something called its prometaphase. This is the phase right in between prophase and metaphase. This is when that nuclear membrane has completely broken down. And then we have the appearance of microtubules. Microtubules are proteins that aid in cell division. And so the most important one is what's known as a kinetochore microtubule. Right in the middle of each of these chromosomes is what's known as a centromere. But each individual chromatid has something that's known as a kinetochore. And a kinetochore is just a little protein that goes and kind of sits in the middle of the chromatid. So the kinetochore microtubules will actually attach to the kinetochore, so eventually we can pull those chromatids apart. We have two more types of microtubules. You have something that's called polar microtubules. That's going to be these guys right in the middle that are overlapping, and their job is simply to keep the poles apart from each other so we can pull those chromosomes apart. And then you have things that are called astral microtubules. These are proteins that, or these protein tubules that will interact with proteins on the cell membrane, and it's also to help keep the poles apart. We then roll into metaphase, which is one of the easiest ones. That's when the chromosomes line up along the middle of the cell or along the equator. Here is a schematic here. You might also see that as a metaphase plate. And then this is going to be a microscopic image of what metaphase would look like. Now we're moving into anaphase, so those sister chromatids are going to be moving away from the middle of the cell or to opposite ends of the pole. Again, schematic here and a microscopic image there. And now we're getting into telophase. This is when we're going to start seeing the formation of nuclear envelopes around the pulled apart chromatids. Those chromosomes will slowly start to become less compact back into chromatid. So again, those beads on a string. And then the spindle will also disappear. So some of those microtubules will disappear. This is going to be a schematic here. What you should notice is that at the end of mitosis, we don't have two cells yet. We have just two nuclei. We now have to go through one more step to finish dividing the cytoplasm. And that's going to be cytokinesis. Cytokinesis is when the cytoplasm actually is divided. And there's something important to understand about animals and plants, and that is that they actually undergo cytokinesis a little bit differently. Plant or animal cells here are going to form something that's called cleavage furrow. So what happens are there's these two proteins, actin and myosin. They're the proteins that are responsible for your muscle movement. They actually form something that's called a contractile ring, or essentially they wrap a ring around the cell cytoplasm and pinch it into two cells. Plant cells are a little bit different. They form something that's called a cell plate. So right after telophase, there are vesicles, and this is pointing to vesicles, that come from the Golgi apparatus that move towards the middle of the cell, very similar to metaphase. And these vesicles contain all of the carbohydrates necessary to produce a cell wall. So remember, a cell wall is made up of cellulose. So all of that cellulose now is forming right in the middle of the cell. One thing I want you guys to think about is why would there be a difference between plants and animal cells when it comes to cytokinesis? And just to wrap up, this is a quick diagram for you. This is for practice, so you should be able to go through this as an onion root tip, and you should be able to identify the different phases: prophase, metaphase, anaphase, telophase. Prometaphase is pretty complicated to observe in these images, but the other four should be fairly straightforward. And this is just one more summary so that you guys can review if you need it.